Anqi Wu is an assistant professor at the School of Computational Science and Engineering within the College of Computing at Georgia Institute of Technology. She received her PhD in computational and quantitative neuroscience from Princeton in 2019. She has a master's degree in computer science from the University of Southern California and a bachelor's in electrical engineering from the Harbin Institute of Technology in China. She's received the USC Chevron Fellowship and was selected for the 2018 MIT Rising Star in EECS. Her research interests are to develop cutting-edge machine learning models for in-depth scientific discovery in neuroscience and build up neuro-inspired artificial intelligence systems, hopefully some of which she's going to tell us about today. And so welcome, Anqi, and please proceed.
Okay, great. Let me share the screen. Okay, can everyone see the presentation slide?
Yep.
Okay, cool. Yeah, thanks, Matthew, for the introduction, and thank you so much for inviting me to present at this seminar. I'm really honored to have the chance to talk about my work here. Again, my name is Anqi Wu. I'm assistant professor at Georgia Tech. So today I'll be happy to have the opportunity to talk my work at the intersection of machine learning and computational neuroscience. The title is Understand the Brain Using Interpretable Machine Learning Models. Okay, so neuroscience have always been studying the brain and also its relations to the input sensory signals and output behaviors. And however, there are some fundamental challenges preventing us from understanding these relations very thoroughly. So here I'm listing a bunch of challenges people have been facing. But fortunately, advanced neural recording technologies have empowered us to dive into our brain and record from the brain at unprecedented scales, and therefore provide us this deluge of neural data and behavioral data for analysis. Although many functions in the brain are still black boxes to us, now we have a large amount of recorded neural activity and behaviors, right? We can develop some artificial models as surrogate models or normative models to mimic these unknown neural functions. And therefore, developing machine learning models is of great significance to really understanding our brain. However, it's non-trivial to develop these machine learning models. We cannot just simply adapt to what we already have in the general machine learning field. We need to bring some neuroscience tailored assumptions to develop a more biologically plausible artificial models. And on the other hand, these models would then be applied back to the brain data to discover interpretable results for neuroscience problems. My work is specifically laid out at this intersection. More specifically, I'm going to talk about my work from three perspectives. The first one is to discover latent structure underlying neural recordings. This focuses on how we should understand the large-scale neural population recordings in the brain without actually getting access to any external information, such as sensory signals or behaviors. And the second is constructing some structured priors for probabilistic decoding for fMRI analysis, where we're also interested in the structures in the brain signals, but meanwhile, we will explore the coding relations with some external information, such as the visual inputs. Lastly, I will switch the gear towards behaviors and introduce some semi-supervised learning models for animal behavior analysis and understanding. The study of behaviors is going to be quite significant to understanding the neural mechanisms and the biological functions in the brain as well. Okay, so let's start from the first one. In a neuroscience experiment, researchers usually have these spectrings like what I'm showing on the right here. We call it high-dimensional spectrum data because there are a large number of neurons being recorded. So by looking at this matrix, it is really hard to tell what these neurons are doing, right? Researchers have been trying to understand the latent dynamics or the structure of these neurons. if we can find some low-dimensional dynamics, we can now explain the behavior of this population of neurons by just inspecting this low-dimensional dynamics. There are already a lot of existing models discovering latent dynamics or manifold like this, so it's definitely not a new modeling topic. But why we still need to build our own model? The reasons are as follows. First, we want to build a generative model for population spectrum data. And beyond that, we need to incorporate some assumptions inspired from neuroscience or tailored for neurodata. And we want to impose explicit manifold assumption over latent structure. And moreover, we care about interpretability of the nonlinear mapping to map from the latent to the observation. We want to model these mapping functions to mimic the functions in real neurons. Okay, so next I'm going to explain our proposed model that will cover these points. Here's the model we propose to solve the task. It is called Latent Manifold Tuning model. Here at the bottom is the spectrum data Y, which is the only observation we have. And our goal is to infer some low dimensional process X underlying Y, as well as some probabilistic mapping PY given X. We first assume X is a random process with a prior PX. One assumption we have is the latent process should evolve smoothly over time, and we can incorporate this assumption into the prior by using a Gaussian process prior. Gaussian process prior can generate some smooth functions. Therefore, using such a prior, X would be a smooth function of time t, which ensures the temporal continuity. Next, we model this probabilistic mapping PY given X via some nonlinear tuning curve F. Tuning curve in neuroscience is usually referred to, is usually used to describe the neural functions between input stimuli and output spike rate. Here, the input stimuli are actually unobserved, which are these latent variables X. So when I say tuning curve, I'm basically meaning this nonlinear mapping function to map the latent X to the neural spectrums Y. Given that, one more assumption we have is that the output of these tuning curves, which are the spike rates, should be non-negative, especially in a spiking model. And another assumption is neurons should fare smoothly over the latent space. Given these two assumptions, we put another Gaussian process prior over the log of tuning curve F for each single neuron. This leads to non-negative smooth tuning curves. So the output from tuning curves are spike rates lambda, which are non-negative continuous values. And then finally, the spike counts Y are generated from the spike rates lambda using Poisson distribution. So this is the generative model we built to discover some low dimensional latent process from high dimensional spectrum data Y. And in practice, we actually reverse the arrow direction and do inference to estimate the latent process and the unknown tuning curves given the observed spectrum data only. Okay. So here I'm showing an application of our model to a rat hippocampal data set. First, I'm showing an example of spectrums for 30 neurons in hippocampus for a 15-minute recording session. These neurons are place cells, which are considered to encode some two-dimensional spatial locations of the rat. Again, by looking at the spectrum matrix, it's hard to tell there's any pattern of neural activity or what's the underlying statistical structure here. If we do principal component analysis PCA on the spectrum data, we will get at least 10 principal components, capturing a significant percent of the total variance. This indicates that the spectrum data has a pretty high dimensional linear subspace, but it is hard to interpret these 10-dimensional subspace. And for our LMT model, we use the test log likelihood to select the best latent dimensionality of the nonlinear model, which falls at 3 here. So this is saying we could find a much lower dimensional nonlinear manifold using our LMT method, which can explain this 30-dimensional original neurospace. So here is how this 3-dimensional trajectory looks like. Note that we actually know that the rat is traversing in a 2-dimensional W-shaped maze while these place cells are being recorded. So we can actually plot the animal's real-time 2-dimensional trajectory here in the middle. We color each arm of the maze for better visualization, and we can see the 3-dimensional latent trajectory recapitulates the behavior of the rat in the physical world while segregating these colored arms. So notice that we actually infer these 3-dimensional latent from the spectrum data alone without the access to the 2D trajectory of the animal. Now, given the 3-dimensional trajectories, we can find a convex hole covering all these three-dimensional curves to formulate a 3-dimensional manifold. This manifold looks like a folded 2D W-shaped maze by gathering the yellow and the green arms to the center, both close to the sign arm. This is different from the spatial relationship between the yellow and green arms. And we also find the same folding phenomenon across different recording sessions and different rats. But traditionally in neuroscience, people usually map the place cells to the 2-dimensional spatial location, right, in the physical world. However, we want to ask, can we find some meaningful information in a higher dimensional space, for example, the 3rd dimension? Here, I'm showing that these place cells actually encode a strong correlation among the 3 arms in the 3rd dimension, which is some interesting structure we find underlying these place cell populations. One hypothesis would be that this strong correlation might be related to the reward delivery at the end of each arm, which is related to some cognitive function instead of just for a spatial navigation purpose. Okay, so this is some interesting thing we can find by inspecting the low 3-dimensional manifold structure. Okay, we've just shown that the manifold looks like a folded 2D shape. And now we want to inspect the tuning curve from the LMT model. And remember that the tuning curve is the nonlinear mapping function to map the latent manifold to the spectrum data. So we could also visualize this mapping function on the manifold for a single place cell. Here we unfold this manifold into a 2D version just for better visualization, which looks like the figure on the left here. It shows that this place cell actually fares the most in the circled region that explain. So basically this is saying this discovered tuning curve can well explain this single neuron's behavior. Here is a quantitative comparison between our latent manifold tuning model and alternatives. We compare the predictive log likelihood on the held-out spectrums. So the higher the bar is, the better. And we can see that our particular formulation pops out as this best model for this specific data. Therefore, it gives us a way to explore a whole class of assumptions about this latent structure given spectrum data alone. Okay, so except for the place cell example, I also use the LMT model to find interesting and interpretable latent structures underlying other cortical areas. Here is an example of visual cortex. I'm not going to dig into details here, but just to show that we can achieve this rotational trajectory as well as the tuning curve for individual neurons on a 3-dimensional torus manifold. Here's another example in the posterior parietal cortex. The mouse executes a perceptual decision-making task with 4 cognitive states, and we discover a 3-dimensional butterfly manifold with 4 wings corresponding to the 4 decision states. And we also obtain the tuning curves for individual neurons on the manifold as well. So all these examples show that our model is capable of finding some interpretable low-dimensional structure to explain the behavior of populations of neurons, but we also inspect the behavior of individual neurons over this low-dimensional manifold. So lastly, I also applied this LMT model to piriform cortex in mice. People don't have much knowledge about olfactory perception because olfactory neurons in piriform cortex don't really have the nice spatial topography as visual neurons. So our goal is to discover some latent olfactory topography underlying these olfactory perception by using calcium imaging data. The data was collected by Bob Dada's lab at Harvard. And in order to analyze these calcium imaging responses, I adopted this LMT model to a new variant to deal with calcium imaging neurosignals instead of spike trains. And the variant will also discover some latent topography instead of latent trajectory with the temporal information as shown before. So here I'm showing the odorant. So before I show the manifold and tuning curve first, tuning curve for this example, I want to show the odorant stimuli first. Our collaborators project thousands of chemical descriptors of odorants down to a 2-dimensional PC space. The first PC corresponds to the increase in carbon chain length, and the second PC separates different functional chemical groups. And the neural responses were collected by presenting a set of 22 odorants to the mice. The set of odorants spans 4 different functional groups, which are indicated by color here. There are esters, aldehydes, ketones, and acids. So within each functional group, the carbon chain length of the molecule is incrementally varied. So this is the topography of the odor set in the chemical space, but it is inaccessible to the modeling procedure. All we have are the neural responses given different orders. So I first applied PCA to the neural response, and here I'm showing the first, the 2-dimensional plot using the first 2 PCs, but these PCs are the principal components of the neural response instead of the chemical descriptors. At the bottom is the LMT estimate. I discovered some interesting latent representations of these odorants, which resemble the true structure in their chemical space. Odorants sharing the same functional group exhibit this line-shaped structure in the data set. And moreover, within each line-shaped structure, the ordering of these odorants is the same as their ordering in the chemical space. Therefore, the x-axis indicates the functional group and the y indicates the carbon chain length. This really provides the nice interpretation for the dimensionality of the latent space that is discovered by LMT. And finally, we can also inspect the tuning curves for individual olfactory neurons to understand how these neurons fare over this latent topography. Okay, so that's basically end of the first part. The summary is the contributions of our work are that we successfully translate neural inspired assumptions into probabilistic modeling. Moreover, more specifically, we propose a generative model for population spectrum data and calcium imaging data. Second, we impose an explicit manifold assumption over the latent process using GP, which is the smoothness assumption in the model. Thirdly, the nonlinear tuning curve we propose here is also more biologically plausible with this smooth assumption compared with an arbitrary nonlinear mapping function, say a deep neural network. Specifically, we emphasize on discovering these interpretable tuning curves to understand how individual neurons fare over the latent space, which is lacking in previous works. And with the model, we extract low-dimensional nonlinear manifolds and smooth tuning curves for place cells in hippocampus, visual cortex, posterior parietal cortex. And I also show an application to piriform cortex with a variant of the LMT model. I find these interesting latent manifold and tuning curves for all these areas. The highlight would be we can discover some interpretable latent structure from high-dimensional neural data alone. Okay, so that's basically the first part about the latent model. I can pause here for a few seconds or a minute in case there are questions attached to this part.
So are there any dying questions from anyone on this first part, or can you leave your questions to the end? Okay, I think proceed, please, Anqi.
Okay, cool, thanks, Matthew. Okay, so far I've talked about learning latent variable models for multi-neuron data. And next, I want to switch our focus from multi-neural recordings to fMRI both signals. There's also some interesting interpretable structure underlying fMRI data that I'm interested in discovering. So I'm going to talk about learning structured priors for fMRI data and how it helps for a brain decoding task. A general problem of interest in fMRI analysis is a regression task, which is usually referred to as reverse inference or brain decoding. We have some experimental variable y as the response. For example, the reaction time during a specific task or the category of represented object. And we want to map the brain activity X, which is the both signal, to the response via some decoding weight W. And we're especially interested in estimating this W. We know that usually, although we obtain this whole brain fMRI data, but only very small regions of the brain are activated in a specific task. Also, another observation we have is that neurons or voxels within a neighborhood should be highly correlated. Therefore, we have the reason to believe that the brain map of the decoding weights W should look like what I'm showing here on the right, with some positive weights, which are these yellow smooth regions, and negative weights, which are these blue smooth regions. The positive weights will indicate the positive relations between voxel activity and task variable. Similarly, negative weights indicate the negative relations likewise. And all the other regions should have no strong activations. So given these observations and the insights about the decoding weights, we want to construct statistical models to estimate W that can take advantage of these regularities. And we built a Bayesian model and assume that W has a structure sparse and smooth prior. Structure sparsity means non-zero decoding weights tend to be nearby each other. This sufficiently reduces a large amount of parameters to fit, similar to like the intuition of using lasso. Smooth means we expect weights within a neighborhood to be correlated with each other. So if we vectorize the brain map of W, the vector should look like this. And our goal is to construct a Bayesian prior that can generate W samples like what I'm showing the right here. So here, I'll explain how we build the prior, but first, let's only consider the structured sparsity. The prior encouraging structure sparsity is what we call dependent relevance determination prior, which is DRD for short. We assume there is a latent variable U, which is unobserved. It is from a Gaussian process prior. As introduced earlier, a Gaussian process is a distribution where you can generate smooth functions from. Therefore, such a GP prior can generate a smooth function U as plotted in the middle here. When the mean of this GP prior is negative and we exponentiate U to get a non-negative signal G, G would have largely zeros, but a few non-zero bumps. Therefore, this G function actually defines this continuous non-zero supports for W. We then formulate a covariance matrix C with G on the diagonal. This is what it looks like. In samples W from a normal prior with such a covariance matrix C, we have structured sparsity. We also then construct a smooth version of DRD prior. Now the covariance matrix not only has this diagonal bumps, but also some off-diagonal values that formulate these blobs for each bump. Samples W from this covariance matrix are both smooth and a structure sparse. Okay, so this is the generative model we built. Again, in practice, we infer the decoding weight W and the unknown latent U given the brain activity X and the experimental variable Y. So here I'm showing an application of the proposed prior to a visual recognition task. The decoding task is to map fMRI data X to the category of the presented stimuli Y, which consists of 8 different types of objects. So it's also considered as a multi-class classification task. We cast it into multiple binary classification problems, and we show here the accuracy performance for all binary class pairs at the bottom. The x-axis is the binary class pairs, and the y-axis is the classification accuracy. We tell that with our priors, we can actually improve the predictive performance for this brain decoding task compared with some competitive alternatives. So this is the quantitative performance. Then what about the decoding weights? Can we qualitatively inspect these W? So here is the brain map of the decoding weight for the house versus a bottle classification task. The smooth DRD weights have significant positive regions in the parahippocampal place area, PPA, which is an area known to respond to images of places. And there are some negative regions in the lateral occipital complex, LOC, which is an area known to respond to objects. By comparison, we can see GraphNet, which is also a famous regularizer encouraging group sparsity, but its weights in LOC are neither strong nor clustered. So this shows that using our smooth DRD prior, we are able to find more interpretable activated RO region of interest in the specific brain decoding task. So to summarize the contributions of this part, I propose a hierarchical model for structured, sparse, and smooth dependencies with Gaussian process. It improves brain decoding with better predictive performance and identifies more interpretable brain maps, and which is the highlight of this part of the talk. Okay, so again, I can pause here for a few seconds maybe in case there's any question related to that.
Again, any burning questions for Anqi on this particular section?
So, hi Anqi, I'm Pan. So let me turn on my video. So I just have a quick question. So do you think the brain, let's say, signal here can be, let's say, we can learn the prior by using maybe some parameterized model instead of like using Gaussian process or something?
Yeah. So the reason we use GP because it's non-parametric, right? We don't want to impose too strong prior assumption to the structure. We just want to impose like smooth or sparse, those kind of thing. But of course, if you have some prior knowledge, like you have some expertise knowledge, you can use that to construct some parameterized prior and to impose that information over the generative model. But generally, people don't use very hard coding, like parameterized form to inform the decoding task. I think mostly people use, even it's parametric, it's more like Gaussian process, Gaussian distribution, or like horse prior distribution for sparsity, like this kind of pretty general prior assumptions, but not very, I'm not sure what the parameterized function that you are referring to, but not likely very complicated.
So I can kind of imagine, so your method is more like, okay, I given one single brain and I given the signal for that, and then I can run this Bayesian network inference and get a parameter there and do this nice analysis. So I was wondering whether, let's say, that kind of prior can be get from a learning perspective. But what do I mean by that? So basically, suppose we observe, let's say, 100 brains, and we can kind of use, let's say, this 100 brains simultaneously, and to, let's say, find a nice prior here, or like a parameterize, I don't know, use neural network or something. So because I guess here, just as you mentioned, I guess when you choose this Gaussian process plus this exponential function, you're also like let's say, impose some kind of assumption or some prior knowledge about the underlying structure. So I was wondering if we have, let's say, more data, whether we can, let's say, find that kind of structure, let's say, in some automatic way. Yeah.
Yeah, that's actually a really good question. And I have one additional slide on this topic, which actually really correlates to what you're talking about. So instead of just impose a naive, like non-informative GP prior over the latent, we can actually construct some prior by learning from the data. And we can say, we can extract some information from the learning process and use that as the prior knowledge for the decoding task. So in this slide, so we have another paper called Brain Kernel, where the high level thing is we want to extract some intrinsic functional connectivity information from a massive resting state fMRI. And this massive resting state fMRI from across a lot of subjects can give us some kind of prior knowledge about what intrinsic functional connectivity would be like in general human brains. And after squeezing those information, we put that into the kernel of the Gaussian process, into the covariance of the Gaussian prior for a similar decoding task. But the hard thing is actually, because there's a lot of misalignment among brains, there's a lot of subjective variability across brains, across different tasks. So how you distill this kind of information and what kind of distilled information you use to promote the performance for what kind of downstream fMRI data analysis is kind of, you have to be cautious about that. You can't just do very arbitrary type of thing and you expect very high performance by doing that. So in this work, I probably won't have time to go through this. It's just basically one slide, but we were pretty honest in the paper saying, even we extract this intrinsic functional connectivity from resting state data and using such information as the prior knowledge for downstream task fMRI analysis, we don't always necessarily improve the predictive performance. And we have to carry out some careful analysis to see why that's the case. Is that because of the misalignment among subjects? Is that because of the subject variability? Or maybe the information we learn from resting fMRI would be no helpful to for the task FMI, saying present visualizing something, a specific object or something like that. So I kind of
can imagine. So because the learning for prior or something like usually they strongly depend on, let's say, some supervised method like labels. So just as you mentioned, so what I learned from you is just like in your domain, maybe the labeling is not like consistent. So we need to, let's say, be conscious of that, or kind of know, okay, what kind of, essentially there are just weak labels and how can we kind of combine them? Maybe it's hard. Thank you so much.
But that's a really good question. Thank you. Since we've been discussing for a while, maybe I should just quickly proceed to the next one.
Yeah, I think so. And then we'll leave final questions at the end. Thanks, Anqi.
Okay, cool. Yeah, okay, great. So far I've talked about discovering structures from neural recordings. However, in many neuroscience experiments, neural activity is highly correlated with behaviors. Therefore, studying behaviors is of great significance to really understanding the neural mechanism and biological functions in the brain. So next, I'm going to switch to the behavior side and talk about some interesting semi-supervised learning algorithms for animal behavior study and analysis. In recent years, there has been an explosion of interest in recording behavioral videos during neuroscience experiments. Often the goal of recording such data is to provide these fruitful descriptors for neurobehavioral analysis. However, feeding models between high-dimensional behavioral data and potentially high-dimensional neural data is a challenging statistical problem. Typically, we want to start by extracting meaningful information from these videos by finding an interpretable low-dimensional representation via algorithms such as post-tracking, video compression, or behavioral segmentation. And then we can decode some meaningful descriptors of behaviors and have more interpretable predictors for neural encoding or decoding models in these downstream analysis. However, when we have very limited amount of human labeled data, we can't really achieve very high quality behavior analysis for downstream application. Therefore, in this part of the talk, I will introduce a series of semi-supervised learning models to resolve this scarce data issue by leveraging both labeled and unlabeled data, and we'll be able to find interpretable low-dimensional representations from behavior videos. So we'll start with tracking first. In a tracking task, we aim at obtaining a bunch of key points capturing the pose of the animal in a video. Animal pose estimation is a critical scientific task in neuroscience. State-of-the-art methods have been proposed to extract behavior traces from different videos, and they all open up an exciting array of new scientific questions and applications to be addressed. A base model that is shared among all these previous models is as follows. Assume we have a label frame. A label frame means we have the ground truth body part locations for this frame, and we input a single frame into a neural network module. This is basically the backbone of pose estimation, like a ResNet, AlexNet. And the output of the network is confidence maps for each key point. The key points here are actually two-dimensional coordinates of the body parts. For example, the body parts pole or the arm in this example. The values of the pixels in these confidence maps are from 0 to 1, indicating how likely these pixels belong to the body part. And moreover, given the two body part locations, we can construct the Gaussian bumps for each key point with the peaks at the true locations. And finally, the loss is constructed to match the output confidence maps from the neural network to the Gaussian bumps from the true locations. However, there are some limitations of such a base model. The first is it only trains on label frames, but much more of the information than these labels about the images could actually be found in many unlabeled frames as well. So we could include unlabeled frames to enhance the training of the network. And second, even with hundreds of labels, users still often see occasional glitches, implying that the tracking might be briefly lost. And these occasional glitches cannot be effectively smoothed out, therefore will usually typically interfere with downstream analysis using this extractive behavior. And our solution is to incorporate temporal and spatial constraints from the behavior videos to smooth out these glitches, but during the training time. So the model we propose is called Deep Graph Pose. It is also a probabilistic graphical model. Let's assume we have a label frame at time t and an unlabeled frame at time t+1, and we can build the same pipeline for the unlabeled frame as well, right? We just don't know the ground truth for the body parts. So here's our solution. We first perform soft argmax with the confidence map to extract a single key point location X and Y. Then we build a unimodel Gaussian from this 2D location given X and Y. We use this unimodel Gaussian as the pseudo key point target map to calculate the loss in between the confidence map and the pseudo Gaussian target map. We call this step as a self-supervised learning step. People might just ask, there seems to be no additional information contained in this Gaussian. But however, via the self-supervised learning procedure, we explicitly encourage the confidence map to be unimodal, which is the additional information we bring to the model. And I will show how critical this unimodal assumption is in the experiment later. The supervised learning and the self-supervised learning parts together formulate the semi-supervised learning framework. Next, we add a spatial constraint to the model. First, let's expand the output to two body parts, which are the pole and the arm in this example. For the label frame, we have both ground truth locations for the two body parts. There's no problem. But for the unlabeled frame, we need to construct the pseudo key point target maps for both body parts using the self-supervised learning procedure. Then we can add a spatial constraint in brain to connect the body parts who really share a skeletal connection in between at each time point. Finally, we add a temporal constraint in blue to enforce temporal continuity of the same body parts across frames. So this is the complete probabilistic graphical model. And with both temporal and spatial constraints, we can smooth out some glitches that break these constraints during training. And moreover, we hope that these additional links we add to the graph could really propagate information from labeled markers to unlabeled markers. Note that the temporal and spatial constraints are not post hoc smoothing, such as using a Kalman filtering. We use these constraints to train a stronger post-estimation algorithm. So here I'm showing the performance on a mouse data set. We are interested in tracking 4 fingers on the left pole. And this data set is a sparsely labeled example with only 55 labeled frames, about 1,000 frames. We compared Deep Lab Cut, which is a fully supervised baseline model, on the left here with our proposed semi-supervised model, Deep Graph Pose. We show that the outputs of DGP are more accurate and robust. To better understand where this difference in performance may come from, we look at this confidence maps from both models for a single middle finger. We can note that the errors in DLC arise from this multimodality of the confidence map, while DGP outputs a unimodal confidence map, which is due to the self-supervised learning framework. And we can see similar comparisons for all 4 fingers. Here I'm showing the comparison quantitatively. We include an intermediate model DGP semi, which is the semi-supervised learning only without any temporal and spatial constraints. The semi-supervised learning improves the performance substantially, especially in this sparsely labeled regime when the training set fraction is very small. And the performance can improve even further when we include the spatial and temporal constraints, as in this DGP full model. Okay, and we also actually developed an extended version of this post-estimation algorithm for multiple animals, which is under review now, that I'm not going to expand too much in this talk. Okay, so I'm done talking about this post-estimation work, and next we want to ask what this tracking information. So here I'm showing that we can actually combine the tracking information we obtain from DGP with a video compression approach to learn interpretable latent representations from behavioral videos. A baseline compression method refers to an auto-encoder, where we input the frames into an encoder, obtain some low-dimensional latents, and reconstruct the frames using another decoder. Obviously, it doesn't require any human label supervision as in tracking. It's a purely unsupervised learning model. But the downside are, the first is the latents are notoriously difficult to interpret. Individual dimensions of latents may represent many aspects of the video simultaneously, and they are all entangled together. Furthermore, the autoencoder doesn't really allow the experimenters to mark features of interest if they want to input something. Therefore, we attempt to combine the strengths of tracking and compression in order to increase the interpretability of the low-dimensional representations. So this is the new video compression model we built, integrating the tracking information from DGP. We introduced this subspace of marker-related latents. These latents encode the input frames and are decoded to generate the markers, which are the X and Y traces in this example. Then presumably, the autoencoder latents should now capture variability in the behavior videos that is not accounted for by the marker-related latents. And finally, we reconstruct the frames from the combined set of latents. Note that this is also a semi-supervised learning framework because we introduce both human label markers into the unsupervised autoencoder framework. So given this model, we could ask how well the marker-related subspace is mapped to the reconstructed frames through this frame decoder. And in this example, the markers are the left pole and the right pole. We first take the dimension, the latent dimension associated with the X value of the left pole, the blue pole. For example, you can think of these blue dots in the latent space to be the X value of the left pole. And we can move these blue dots up and down, but keeping all the other values fixed. We observe that our model has learned a single latent dimension that is mapped to the horizontal movement of the left pole. Importantly, there's no vertical movement of this left pole, nor any movement of the right pole. All of that information should be contained in other latent dimensions. We call this phenomenon to be disentanglement. And we can do similar manipulation of other latents in this marker-related subspace, and we see similar disentanglement across different dimensions. Similarly, we can ask how well the autoencoder subspace is mapped to the reconstructed frames, which will also give us some idea of what information that subspace contains. And for the first latent, you can clearly see the draw opening and the town extending, while other aspects of the image, notably the pole position, stay constant. The second latent corresponds to local movements of the pole, but along the base of the holder, most likely capturing some small movements that is not accounted for in the tracking information. So, okay, we're able now to interpret these auto-encoder latents, then what we can do with these latents. We can segment just a subregion of a video. We can decode individual dimensions of behavior, and we can have more interpretable predictors for neural decoding models for downstream analysis. We actually have already done this further analysis regarding behavioral segmentation and neural decoding of interpretable behavioral latents. So please refer to the bottom paper here for more details. Finally, I just want to briefly mention that we also have been working on some semi-supervised learning model for behavioral segmentation. But again, please check out the paper here for more details. Okay, to sum up, the contributions of these projects are, we propose a new probabilistic graphical model for post-tracking first. We also propose a new video compression method with the DGP tracking information. These models are both semi-supervised learning frameworks that can leverage both labeled and unlabeled information. And most importantly, they both render us these interpretable low-dimensional representations of behaviors that are really beneficial for downstream neural behavioral studies. Okay, finally, just to remind, this is an outline of 3 topics that I've talked about in this presentation. As a consequence of the fast development of recording technologies in neuroscience, we are obtaining more and more complex neural and behavioral data nowadays, which will be a norm in the future. And we need to break down these 2 very complicated domains and marry them in a very efficient and impactful way. We could really deal with such problems for both system neuroscience with animal study and cognitive neuroscience to study humans. With all this complex data collected, I've shown how to analyze them by building different probabilistic models and the inferential techniques. Moreover, there are all sorts of places where computational approaches can really make serious contributions to study the brain. And furthermore, there also exists an inspiring direction to develop a neural-inspired AI by bringing the knowledge we occur from neuroscience to build more interpretable artificial models. So there are plenty of interdisciplinary research opportunities between neuroscience and artificial intelligence that I'm very excited about. And there's also a huge need of researchers to get involved in. Okay, last but not least, I want to thank all of my experimental and scientific collaborators. I'll stop here and welcome any more questions. Thanks for listening.
Thank you very much, Anqi. That was extremely interesting. So we have time for some questions. Please put your hand up if you have a question. Do we have any questions? So let me ask a question then from the first part of your talk. I was just curious about the, you're inferring these low latent dimension structures, which seem to be having some mapping into the actual spatial orientation of what the animal is doing or such. Is that just coincidental, or do we actually sort of think that there is some sort of real spatial representation of the activity in the brain relative to what the spatial orientation of the behavior might be?
Yeah, that is true. So place cells in the first example, right, I'm showing the latent model analysis of place cells. And place cells are well known to encode to the spatial locations, and they're the most important cells for spatial navigations. So basically, animals encode the locations of where it is using all these place cells. And if you, so basically the whole population will encode a full spatial mapping of the animal. So that is anticipated, and that has been well studied. And therefore, it's actually, that example is not some novel discovery using our model. It's more like a proof of concept.
Yeah,
because
yeah,
we're expecting that. But a bit new thing we did beyond that, the discovery of 2D spatial location, is we found a third dimension. And this third dimension actually incorporates some reward related stuff. And reward delivery is not just spatial navigation, it's related to cognitive function. So the animals, that implies that the place cells are not really just encoding spatial locations, but also some cognitive functions related to reward delivery. So people have studied place cells for spatial navigations for years, and people have already also extended to higher dimensions to see what other information place cells can encode. So that's very active research topic in the field. Cool.
Yeah. Well, I'm an astronomer, so I know nothing about how the brain.
Oh, okay. Yeah. Okay.
Such is the nature of A3D3. But yeah, oh, thanks. That clears that up. Yeah, that sounds very interesting.
Okay, thanks for a question.
Any other questions from anyone? Eli?
Yeah. Hi, Anqi. I first wanted to thank you for a great talk and visiting A3D3 seminar virtually. It's really great to hear all the collection of works and how it is related to different problems in neuroscience. Second, I had a quick question to kind of like try to understand how the first method that you were showing, the LMT method, how does it compare in terms of accuracy of prediction of the neural time series versus those methods that don't kind of like require a particular low dimensional latent representation?
So
let's say like LFADs or similar kind of methods to LFADs.
Yeah, so we didn't really compare with LFADs in the paper, but we did pair with other some pretty popular alternatives like Poisson LDS or Poisson GPFA. These competitive models basically try to discover a similar thing, but they use different model assumptions. For example, Poisson GPFA, they don't have a nonlinear mapping or nonlinear tuning curve, but we assume this nonlinear tuning curves, which are generative from another Gaussian process. So basically, we compare with all these methods and show that we do have pretty high predictive log likelihood when we predict for new time series spectrum data. Alpha, I didn't include that into the comparison back in 2017 at that time. So, but it would be a really interesting model to compare against, definitely.
Yeah, my question stems from this attempt to understand whether if you are trying to find out the most optimal representation, whether you pay some price in terms of kind of like the prediction and the reconstruction of the signal.
You mean sorry, can you repeat the first part of the question? That you would pay the price for what?
Yeah, so if you are looking for like this low dimensional, really interpretable representation, whether the accuracy of reconstructing the signal or predicting the signal is going down or actually improving.
Yeah, actually, I think we want both interpretability and predictive performance. So generally in the field, if you assume some low dimensional manifold or latent dynamic structure, the performance, like the generalization performance, is usually improved. If you want to reconstruct the same training data, you easily get overfitting, right? You use all the neurons. But if you kind of go down to a lower dimensional subspace and do prediction for unknown time points for new data, the generalization performance is usually improved. even not using our model, like using Poisson GPLA or using LFAS or using PLDS, the performance is improved. People also compare these methods to pretty simple like PSTH or very simple smoothing function methods, and they show all significant improvement over simple like statistical methods without a low dimensional assumption.
Great.
Will that help? Okay.
Okay, so I think we're just past the hour. So thank you very much again, Anqi, for the talk. And everyone, please bear in mind that the next A3D3 seminar will be the first Monday in May. And it's going to be from the heterogeneous systems group. Okay, thanks everyone.
Yeah, thank you again for inviting me. I'm really happy to have the chance to meet you all. Thank you.
Great.